my great pleasure to have Luca here. So Luca works at EMBL Heidelberg in the Oliver Stegle Lab. And I don't remember, I think we somehow met or I got to know that you guys are doing really a lot of awesome stuff with spatial transcriptomics data, and somehow then we thought and you were already using ZARR-based I think something, right, to save the data, and then we thought, wouldn't it be great if you try to adopt or contribute to the OME-ZARR standard so that we can then all read and write the same formats? And judging from the title of your talk there, you adopted this idea. So this is awesome. That really makes me happy. So yeah, looking forward to your work.
Yeah, thank you. Correct. We were using HDF5, but now we're using ZARR, as you said, and we'll go in all the details very soon. So I start sharing the screen.
Yes, we can see it.
Thank you. 1 second, I move one Zoom window. Okay. So it's my pleasure to be here today talking about the work that I carry on along with the members of my team, some teams outside the institute. And we work with spatial multi-omics. We try to propose a solution to some problems that researchers face every day with spatial multi-omics. And for doing this, as Christian said, we adopted some ideas and solutions from the imaging world. So I want to start with an introduction to spatial multi-omics technologies, in the case in which some of you are not familiar with, and also take advantage of this introduction for explaining the problems that arises. So the formal definition of a spatial technology is a technique, an experimental technique that allows for the detection simultaneously of quantitative information along with the positional information. An example of a technique that is not a spatial technology is single-cell RNA-seq, because this doesn't give any positional information once the tissue is dissociated. And on the other spectrum, to be very strict, in situ hybridization with bright field microscopy is not a spatial technology because it's not quantitative. It's difficult to compare in a very defined way numbers between different areas of the same samples, or even worse, different samples. So what's in between are is a long list of spatial methods. There is almost one method for each letter of the alphabet, and this list is keep growing. Along with these techniques comes the first problem that often each technology has got this raw data format, but even more important, the process data changes in the way it's presented depending on analysis and also the format that it comes in the beginning. So now I want to put some order in this list of methods, and in my team of work, we identified a classification of these spatial technologies, and we propose that you can see this big list as these three groups. First, there are annotated regions, and these are 2D or even 3D areas, regions, volumes that can be annotated with an expression or some categorical information. The most common example is Vision from 10x, but there are different techniques that we believe fall into this category, like a spatial transcriptomic, in which the circular areas are larger and the disposition is a checkerboard-like grid. Slide-seq, smaller areas, random disposition, but also things like Tomoseq, in which there is an organism which is sliced in consecutive slices, and each slice is one sequence. So now the region is the slice. The second group is a single molecule, and here we don't have areas, we have points. Each point is a single molecule, is a transcript or a protein, and there are different methods that gives this. And the last one that we identified is multi-channel high-resolution raster images. Raster because this is a raster information. And here, even if I said that strictly, microscopy is not a spatial technology, it's convenient for a practical perspective to put also microscopy images inside here. And now I want to show you for each of them a bit more in detail one representative technology, but most importantly, I want to show you how the data, the raw data, is stored and how the process data looks like in a real data set. And sorry, yeah, if it can be a bit confusing. So if I just want to say, if you lose like track of all the details, it's not a problem. This is exactly what we try to solve, and then we will show a solution for all of this, let's say, messy way to store the data. So the Vision technology works by placing samples on a slide containing some circular capture regions. They are also called in jargon spots. Then the tissue is made permeable, and then the transcript bind to the primers that are spatially barcoded, and then you can proceed with Illumina sequencing to find sequencing information along with the region they come from. The spot is 55 micrometers in diameter. This is not a single cell technology. This is taking some of different cells. And the data looks like on the left. So we have an image in the background, and then we have this circular spot that are annotated with an expression information. The data, this is a real data set. The data may look like this. And here in green, I put what is raw data, or like closer to the raw form of the data. And then in black, there is process data. And in a spatial representation, we want all of this. But let's focus on the green one. The green files are the one that allows you to reproduce the plots on the top right. So the first two files are an expression matrix and a list of positions that allows you to make the plot on the left. And then there are two more files that are images of different resolutions that allows you to make the plot on the right. Finally, there is another file which contains some scaling factors allowing you to map the coordinates of the plot on the left to the plot on the right. And the representation is actually really diverse for this kind of data within each other, because some matrices are a sparse metric representation stored in the SDF5 format. Other ones are in the CSV format. The scaling factor are in JSON format with these conventional namings. But then if you look at the process data, you can get really heterogeneous, because here we want to save clustering information, embeddings, output of different tools, and there can be really everything. The second example I want to show you, the second category, is a MERFISH dataset, and the data look like this. We have information that we can zoom in until we get a very fine-grained subcellular resolution, and each point is a single molecule. And the experimental methods, just also to get you a bit more familiar with the data, is a very simple idea. You have some targets, and you put some fluorescent labeled probes inside the sample so that they bind the target, and then with fluorescent microscopy, you can see these bright spots. And by making many probes per target, you get a very bright spot that you can identify as a single point. This doesn't scale to many genes, so you actually introduce an artificial barcode. So you do staining and then probe removal, you rehybridize, you remove again the probes. So for each location, you have a sequence of colors, but then this is the very raw data. And then what you do is that you find the coordinates and you assign a gene for each coordinate. So this is how the techniques work. This is instead how the data look like. So in this dataset, this is a mouse brain MERFISH dataset. There is a data frame stored in a CSV format in which you can basically produce the plot on the bottom right. I actually added these large color circles just to give you a reference for the single cells, but the information is just this list of single molecules. And for this single cell information, I actually use another file which is present in this dataset, which is a set of circles of different areas showing where the cells are located. And also we have an expression matrix. So we have the genes present in the MERFISH dataset are accumulated into these regions by summing the number of coordinates, number of points inside each region. So this information is not raw, it's like deduced from the MERFISH data, but is very close to the raw information. It's like one of the first steps you do if you work with this data. Then in this dataset, there is another file, which is a polygonal description of some larger anatomical regions for the layers of the visual cortex of this mouse brain. And in this case, this is saved in a GeoJSON format. Finally, last technique I want to show you is imaging mass cytometry, called also IMC or commercially Hyperion. And this works by staining simultaneously both RNAs and proteins. And then with a laser, you pass every position in this sample and you destroy its point, what is produced from this operation is passed to a mass cytometer, deconvolving what's inside, and you obtain high resolution multi-channel images. The data, real data set, this breast cancer data set from the University of Zurich, look like this. Again, there are a lot of process files, but let's focus just on the raw data. And the raw data are just images, multi-channel images, but very close to the raw data generation step, there is actually the identification of single cells. So you, the researchers run instant segmentation algorithm to find where the cells are located. So you have these raster masks, these instant segmentation masks, and then they counted the pixels, they summed the pixel intensities for each cell, so they obtained also spatial single cell expression profiles. So this is the raw data how it's stored. And again, then there is a lot of process data like square waves, cell distances representing graphs, boundaries of polygons, embeddings, channel information, and so on. So this was the last example of an IMC. And a warning, if you have another IMC dataset, it doesn't mean that it will look like this. There is another IMC dataset that I work with, and the representation was different. So I think this really shows the first problem that I mentioned, technology-dependent formats for storing raw and processed spatial data. The researchers, to address this problem, but also to be able to analyze the data, introduced some framework for spatial omics analysis. And this framework introduced an ad hoc memory or a storage format, and they can be used for analysis and plottings. The frameworks are very valuable, and they contain also valuable components that you would like maybe as a researcher to reuse in another context, because different frameworks do different, slightly different things. But then there is a second problem, that the different data representation that they introduce links to a difficult reuse of the frameworks across each other. On top of this, there is a landscape of single tool tools, and each of them has got an ad hoc input because they are designed to be technology specific, like this tool is designed to work for Virshup data. And then also sometimes they are language specific, like they are written in R and they work with Python, so there is not an easy crosstalk. So this is our second problem, different non-interoperable frameworks or tools for spatial data. The main frameworks that are available around are the one that I mentioned, that I show here. And I want to focus on a few points here. The first one, the two columns on the right, is that despite this tool being highly valuable, unfortunately, no tool works with IMC data using like instant segmentation information. If you want to work with IMC data, you have to use a different tool that is just designed for IMC data. It's called IMC Toolkit. And also, they don't support multi-modal data. Or to be more precise, if you pass me the term, you can work with multimodal data with Aki way, but not in a structured way. And the next slide will see in more formal term what I just said. But regarding the analysis, for instance, I work with Python. Sometimes I would like to use Zoto or Sura, but I'm not doing because I have to convert the data, and that's very time consuming. Or with Zoto, there is a very powerful interactive viewer that I would like to use more often, but then it's really painful to convert working with the Python language. And the last point I want to stress about is this X here, technology-independent storage. With Sura, unfortunately, the classes are designed in this way, so that we have a class for the main spatial technologies like Virshup versus one or slide-seq. And this creates, I think, a problem in extending the methods to different technologies, because Virshup and slide-seq are very similar. What changes is the spot size and the disposition of the grid, but when the classes, like in this example, are technology specific, then this hamper data reuse. So talking about spatial multi-omics, which was the last column on these tools here, I want to show you a solution from the non-spatial world, because the problem was present, of course, also with standard single-cell technologies, but a solution has been proposed. There is a solution in R. I will show the Python solution here. I'm more familiar with this. And for single modality data, you may be familiar with EMData. EMData is a representation of matrices that comes along with annotations. And there are two concepts, the concept of observations, OBS, that can be like cells, regions, samples, and the concept of variables, VAR, that can be genes, proteins, features, and so on. And this EMData contains annotation both for OBS and for VAR for a single modality. Extending this to multimodality required a different design, and new data was recently being proposed, as a way to encapsulate multiple M data single modality objects in a larger containers that inherits all the advantages of M data, but allows you to store annotations and metadata also at the outer level of the hierarchy. So now you can have modality level information and also global information. And data and data, which are interoperable, are bundled into an ecosystem called the SCVerse that encompasses some core tools and then many third-party tools that allows to build on top of this data storage. So now the data storage for multi-omics data is not going to be a problem anymore. This is what the idea that we want also to apply for spatial data. So we have the third problem that I explicit here. Solution exists for non-spatial multimodal data, but not for spatial multimodal data. So now that I show you this, I want to show you the solutions that we propose. And the solutions are in terms of 3 Python packages. And let's start with the first one. The first one starts by first introducing an abstraction in how the data is represented. So I mentioned before that you can classify spatial information into these 3 categories: annotated regions like Visium, single molecule like MERFISH, and then multi-channel images like microscopy or imaging mass cytometry. So what we see is that each of these groups is basically one single object. We don't want to introduce different classes for different technologies. We want to introduce these 3 building blocks, named annotated regions, single molecule, and raster. The advantage of this is that if I mentioned before that Visium belongs to the annotated regions, is that if you have new spatial data types, like when you get Visium data, it's not like Visium, is called Visium ACNI, and Visium ACNI is just Visium expression matrices plus images, is what I show you in the slide before. And this is not one single building block, is a combination of building block, is like regions plus raster, but so is also like nanostring data. Nanostring is a background fluorescent microscopy image plus some regions. In this case, I'm showing some isocontour curves that are determined from the boundary between a healthy and tumor tissue. And from an abstract perspective, this is just some geometrically described regions plus some images in the background. So the advantage is not just in data representation, but also in tool repurposing, because if you have a method that works for Visium ACNI, sometimes, not always, the methods actually can be abstracted as working with a combination of these 2 building blocks, regions plus raster. And when this is the case, then you can apply the method to nanostring data. It doesn't stop here, because as you can combine multiple building blocks to represent new spatial technologies, you can also combine multiple building blocks to represent a spatial multi-omics dataset. A spatial multi-omics dataset looks like what I'm going to show you in this slide. So usually you have a sample, and then you introduce some consecutive slices of this sample that can be of different thickness, and then for each of them you use a spatial method. The topology of a sample can be slightly different because of natural deformation of a sample, but is more or less the same, and you can proceed with different spatial or even sometimes non-spatial data for the analysis. So this is an example that will be recurrent to the representation of a spatial multi-omics dataset with 3 aligned layers. There is a microscopy layer, there is a Visium, purely Visium expression layer, and then there is a MERFISH layer. So this is a raster object, this is a region object, and the top one is a single molecule object. And these layers, the key is that they are spatially aligned. So the same area in space can be analyzed with different technologies, looking at the different modalities of the different layers. So how to store this information? The first thing, we need to store each block, but also we need the block to be related between each other and related between the physical space. And luckily, from the imaging world, the problem has already been addressed and already been solved. So what I'm showing you here is the specification for OME-NGFF, which is a community-driven standard for microscopy data. And there are implementations based on OME-ZARR that take advantage of cloud support or chunk storage, and also support for multi-scale images. So it works like Google Maps. You can zoom in and then access targeted storage chunks instead of having a big image that you want to load, that you have to load in the beginning. So this format is basically storing images and then storing labels. Labels are segmentation masks, are images of the same resolution as the microscopy images, but containing integers with the label information about the cells. On top of this, as I said, there are coordinate transformations, there are translations and scaling factors, and also the mapping with the physical space with, for each channel of its image, there is a unit, like a unit of space, micrometers, or a unit of time. And there is an ecosystem built on top of this, there is, as I said, a ZARR implementation, there are libraries for input-output, and also you can use tools like Dask for lazy loading and parallel processing. So this looked like a very good building foundation for our representation, but we needed extra storage information. In particular, for representing spatial multi-omics data, you need a representation for single molecule and a representation for regions. These regions, or to be precise, annotated regions, are basically geometrical description of the regions plus the annotations. The annotation is simply a non-spatial and data object, and the unannotated regions, the geometrical description, can be done in different ways. So here I'm showing you that if you want to represent some circles, you can have a center and a radius. You can have a raster set of masks, rasterized version of this circle, or you can have some polygonal description. And among all these building blocks, the raster and the raster regions are supported by OME-ZFF already. The other one not. So what we are doing in the team is that we are proposing in the repository in GitHub some new specifications and discussing them to be able to encompass this. So this is at a glance what the new representation will look like. The one on the left is the already present one with raster support and the raster regions support. And the one on the right is instead of labels, is introducing this concept of regions. So these regions can be defined as labels, segmentation masks, but also as geometrical description. So with like center and radiuses or polygons. And then on top of this, we will need some new types. So types for representing single molecule data. Also, we're exploring quad trees or octrees for a better indexing. And then also non-spatial annotation tables. And these will annotate the regions, for instance, but also we believe that it will be useful to have annotations at outer levels of hierarchy. Because if you have multiple images, maybe you have tables that have one row for each sample, not one row for each cell. So this is, in short, the specification, but this is just the file storage. We need in-memory representation. So our Python package, we call it spatial data, allows you to do input-output and represent this format in Python, but also to do some basic manipulation. You want the object to be able to modify itself, still having minimal dependencies, Python dependencies. So this, let's see an example of these manipulations. So let's consider the example I showed you before of these 3 aligned layers of different building blocks. And the spatial data format doesn't just limit to 3. As a user, you can stack on top as many aligned layers as you want. You can decide to split them in memory into different blocks, and also you can decide them to split them into different files. So you're not forced with one single monolithic object containing everything. You can take it, rearrange it as you want, split it, merge it back, and it's possible to do this because all the blocks are aligned in the physical space. So it doesn't really matter how they are stored in the end. This alignment allows you this kind of operation. So this an example of a local coordinate. You have a local coordinate space that can be like the space of the image, in which the numbers in the axis are pixels, but then you want to map it in the physical space, and you may need to translate it, rotate it, and even stretch it. And then you have also mapping between these local coordinates, these pixels, and the physical space with like micrometers. So these kind of transformations are stored in this format. You then, from the memory representation, we provide different building blocks for its different classes, different Python classes for its building block. And for doing this, since we don't want to re-implement the wheel, we use existing standard technologies. Like for images, we rely on Dask for lazy loading. For a features table, we make use of mdata, and for regions, we use a combination of mdata objects and spatial indexes objects, like we are exploring, as I said, the use of quad trees. These building blocks are designed to fit the needs of the user and not force the user into a particular way of thinking. And I explain this better. Sometimes the information that you want to store is not, there is not a canonical way to store it. As I showed before, there are sometimes some alternative ways of representing the same information. Sometimes the conversion between these building blocks is lossy, but it can be like a price that the user is willing to pay for a more convenient representation. So we provide methods for going back and forth this representation. This is not the only case in which the tool is doing this for the user. Also, when you have a high resolution image, you may want a lower resolution image. You want to do downscale. And when you downscale, the problem is that the coordinate transformations need to be updated. And right now you have to do it manually. So the tool does that for you. So if you do a downscaling, the coordinates are under the hood computed and saved, and you don't have to worry about this. A more complex, a more advanced example of equivalent representation is in this case between spatial transcriptomics data and an alternative representation for it. So spatial transcriptomics is the precursor of Visium. You have regions, annotated regions that are disposed in a checkboard-like grid. Sometimes it's convenient to see this as a multi-channel image. And then maybe you want to go back and forth this representation. So this is an example in which our flexible combination of building blocks allows the user to do this. And the last example I want to show you of, again, going back and forth these building blocks, when you have single molecule data, you can have millions of points. And if you want to apply like deep learning methods, but also like for manipulation and plotting, is not the most convenient form. You don't want to pass millions of points to your neural network. So a step can be rasterize the data. So it's basically introduced a set of squares in a grid and then counting the number of molecules inside. And this is what the rasterization looks like in this case. There are already methods for doing this, like the library data shader. But there is the same problem as before, that when you do this, you need to recompute the transformations. And the methods does that for you and saves them for you in the data storage. So this was for going back and forth between different spatial building blocks. Sometimes you start with spatial data and you need to obtain a non-spatial summary, a matrix of features that then you can process with non-spatial methods. And we already saw an example of this before in the introduction, which was when you have single molecule data and you have some regions. And then you want to count the number of points inside each circular region, and you obtain a matrix, a feature matrix. So this is an operation that we support, but we support it also more generally. You can use a different building block type for the source, like you can use an image, and you can sum pixel intensity inside the regions. But also for the regions, as I said, we support different representations. So it doesn't really matter that you have like shapes or segmentation masks. Under the hood, the API will resolve the query in the way that has been asked for. And a more complex example is that you can also actually change the source, and you can also use the regions as a source to aggregate into other regions. And I show an example of what I mean. You can have as a source some Visium regions, some circular spots, and then you can have on the right an anatomical description of anatomical regions, larger regions. So this in a case which you take the Allen Brain Atlas and then you register it to your data. Maybe you want to aggregate the Visium information on top of these larger regions. And again, from our perspective, this is still the same query, is accumulate. And it doesn't really matter what the source, what's the target, the method does that for you. So all these operations were possible because layers are aligned in space. Alignment in space allows you also to do cropping operations. So you can have a large data set, but you are interested because maybe you want to show a plot to a collaborator just to a small subset. So you can select maybe in the physical space a region, and the method will crop each layer in the original space for you. And you can also do this by specifying physical coordinates. So if you have an image, instead of saying, give me a crop of 1000 pixels, you can say, give me a crop of 100 micrometers. And the methods will get the pixels that are needed for that. And you can also go in the other direction. So you can go both from a physical space to the local space, but also from the local space to a physical space. And this is very useful if you do deep learning, because if you have like a Visium dataset, you may want to apply a ResNet, a convolutional neural network, for its image style around one spot. And this kind of query is made possible by the library, because you can say, I want all the tiles of 55 micrometers around each Visium circular region. And this is what the method gives you. Spatial data will have Python generators that can be used to be plugged inside deep learning and statistical learning frameworks like PyTorch and TensorFlow. The last few features, and then I sum up, is that this is an important point. We put the priority into alignment in space. So if you consider to have two layers of the same information, like you take a Visium layer and you duplicate, you need to know which spots match in one layer to the other. And imagine that you take a clone of this. So like the top left spot will be the top left spot in the other layer. But then you do some processing. So like you filter out some spots in one layer and some other spots in another layer. If you then rely of ordering to match the coordinates, you have a mismatch in the spots. If you rely on a unique identifier, it works. And then you have to be very careful that you have different layers that you are using consistent indexes. So our method allows for convenience reindexing based just on spatial overlap. So you can just take as input these two filtered layers and say, give me the new indexes so that they match in different layers. And there are cases in which this is crucial. So this is actually a real example from an analysis that I've done before developing this method, with I had some IMC data and I had different instance segmentations of the same information. So like this was algorithm 1 and this was another algorithm for instance segmentation. So if you look at this closely, the regions mostly overlap, but some of them, they are split into different cells and they are also not pixel-wise overlapping. In this case, matching instances can be really tedious, and we are designing a method that does this for you based on spatial overlap. So this was an overview of features. I want to conclude with two examples of how analysis will look like using this method. So let's again consider our spatial multi-omics dataset, and let's save it this time into 2 layers instead of 3 layers. Let's say that there is the Visium ASNI layer and the MERFISH layer. It's up to the user to choose this hierarchical categorization. So the raw information that you see on the left is stored in this way. You have the raw expression from the genes, you have the low resolution image, and then you have the MERFISH single molecule information. The first thing you want to do is applying some preprocessing. So there are already tools like ScanPy or SweetPy that allows you to filter genes and then compute clustering and embeddings, and you can save this inside our representation, inside this mdata object. And then you can also have a larger image that you may want to align. So we have methods to align large images and small images with like 1 line of code and then store them aligned. You can apply, let's say, an external tools to this high resolution microscopy image for finding cell nuclei, and you get some segmentation mask, and you don't have to worry about where to store it because you can just store it inside our storage format. And then looking about the multi-omics part, you can use these newly computed segmentation masks to count the number of molecules inside each region from the other layer, multi-omics layer. The second example is actually not from spatial multi-omics, it's from organoid, working with organoids. So let's suppose that you have one sample, one organoid, and inside here you can identify some cells and you can compute some features like shape of the cells and other image features based on this microscope image. And let's suppose that you have different organoids, like 100 organoids. So you have many little feature matrices. One step is to merge all these feature matrices, and then maybe you apply some operations on this large matrix, like you lead to some row removals. And we support both tables at the local level, but also at, as I said, outer level of the hierarchy. So you can have a global table that maps to local samples, so that if you remove rows, then we take care of working with indexes so that you can map this new information back into space without having to remember which rows were removed from which samples and so on. So this was all. This was spatial data, the packs that we are developing for storing and manipulating spatial multi-omics data with our building block abstraction. And now I want to show you the other two packages. So the second package is a purely visualization plugin. This is an Napari plugin that is extended to work with spatial data. Giovanni and Isaac and Kevin contributed in SquidPy and Napari, and all these three people are in the team working on these projects. And in particular, Giovanni and Isaac developed an Napari plugin for SquidPy that is the one I'm showing you in this slide, that allows you to show the SquidPy spatial information. This is stored as an mdata object with some extra coordinates. And you can see genes, you can see cell types, and you can also, inside the Napari interface, select some polygonal regions, and Napari for you knows which points are inside this polygon and saves the annotation. It doesn't save the regions, it saves the annotation, saves this orange versus blue. You lose the information about this polygonal region. So what we want to do is to extend this to spatial data so that Napari is actually working with the spatial object and is supporting also all the methods that were developed before. So it will also interface with SquidPy. And on top of this, we have extra features. So not just interactive visualization, but also sample comparison in the case which you have organoids, and better bidirectional communication. So you can define regions and save them in the spatial data object. You can also align layers. You can do some registrations within the power of Napari. So we have a demo of this. This is actually not about interactive operations, but only visualization. So I want to quickly show you. This is the Merfish brain dataset that I showed you before in the introduction. So we have an OME-ZFF OME-ZARR object containing the single cell circles, and we can plot them with Napari. We can zoom in, and we can also plot the expression on top of this. So we have an annotated table. This is basically an mdata object saved to OME-ZARR, and we can use our tool to plot it on top of the regions. We can show the single molecule data. This is an example of the single molecule data aligned to the larger regions. And also we rasterize the single molecule data with data shader. And we have this new alignment. This requires a new alignment that is computed and saved into the format. So now we can plot the single molecule data and the other layers on top of this rasterized information. We also have in this dataset some polygonal descriptions of the anatomical layers of the mouse brain, and this is saved and can be plotted again. And also we showed this also for vision data, and I want to show you also this for imaging mass cytometry data, where we have some segmentation masks that are saved as labeled images, and we can put some annotation on top of this. And this red cell here is came out because the annotation were actually mismatching the original pixel regions. So the method is designed also to work with these cases in which the data can be a bit corrupted, but still you are plotting the right expression on top of the right cell. So let's go back to the presentation. And let's show, I think I have 8 minutes, the last package, which is SPIN. So if you look at the non-spatial world, you have a separation between storage and analysis. You have mdata for storing information and then scanpy for analyzing the data. And data is very lightweight. You can import it easily in your Python environment, and then this led to other tools adopting NData as the way to store spatial information, to store non-spatial information. So with spatial data, we decided to do the same. So we are developing analysis package called SPIN, spatial analysis. And the idea is that since spatial data will be lightweight, you can, as a method developer or as a user, use spatial data without having to rely on an analysis-heavy framework. And the SPIN has got three purposes. The first one is implement single-modality methods, in particular, all those preprocessing methods that or analysis method with complex dependencies that you don't want to include in a basic data package. And SquidPy already implements many methods for single modalities, so SPIN is compatible with SquidPy. And an example that refers to a previous slide is when you have Virshup dataset, you want to apply some deep learning methods. So with spatial data, you can generate these crops, but then with SPIN, you can actually construct, for instance, PyTorch data loader, and then you can apply a pre-trained neural network, you extract features, and then you can go back to spatial data to plot information from a neural network. Like in this case, I'm showing an idea of how activations from the last layer look like. So the second part of SPIN is the crosstalk with external tools. As I said, there are many analysis tools that we want to reuse. So we have the objective of supporting importers and exporters for this, and also have convenience bindings to external tools, so that you can call an external tool within the SPIN framework. We are exploring an optional part of using Dockerized callers, so that you can also call, let's say, from Python, tools from R without having to worry about the environment. And this is an example from spatial data. You may want to convert to Giotto, to then use Giotto, but you may want also to use, without having to create explicitly an intermediate Giotto file, to use the Giotto interactive viewer within SPIN. So we are exploring this link with Docker. Finally, last point is about multi-omics. There are already tools that accept some multi-modal input, and we are finding them and repurposing them using our building block abstraction. So an example is a method from last year. It's called XFuse. This allows you to give input as input the layers on the left. This is a spatial transcriptomic data set and a microscopy image. And this is basically creating a high resolution spatial image. Is using deep learning, holistic deep learning is creating this inferred representation. And with our abstraction, we can say this is a combination of a region object and a raster object, and gives us output a raster object. So you could apply this also to nanostring data. I have the last few minutes, so I want to show you quickly a code demo. Christian, how many minutes do I have? Because I may skip some slide.
Yeah, maybe come to the end that we have a bit of time for discussion if possible.
Okay, yeah. So I just show you maybe the first and the last slide of the code demo, and then we can discuss later. The demo that I show you is actually so the method that I show you born in this way. In my lab, Danila and Ilia developed Nuon, and then after we developed this for a non-spatial data, together with Ilia, we explored a prototype for spatial data. So we developed a HDF5-based storage that was having both analysis and storage together. But then when we saw that people were happy about this and it was working, we decided to restructure this and join forces with external tools. So now we are developing spatial data and SPIN. But this means that the spatial data and SPIN code is not ready yet. And the demo is from the old spatial muon. So this is just to explain why you will see spatial muon instead of spatial data. So spatial muon allows you from a command line or from a Python interface to look inside this object. In this case, as I said, is a HDF5 storage. And this is very similar to what I showed you before. So you can see the many layers inside the object with the spatial information. You can plot them, and you can also combine the plot together because the layers are spatially aligned. And then, I'm sorry, I will skip some slides and I will just go to the interesting part. So if you have some Virshup data, you can apply deep learning. So with our method, you can have small resolution images and high resolution images, and to align them together is basically this just the code that you see up here. So you have the function align, and computes for you the affinity transformation to map the large image into the small one. And then we have our methods for generating tiles for deep learning based on the masks. So you can generate the tiles and also map them back to space. So I use this to develop a deep learning method for spatial multi-omics, and I was able to apply it both for Visium data, these are the crops from the Visium data, and imaging mass cytometry data using the same code, even if the data representation is very diverse. And there are already tools doing this, like PubML or SquidPy, and the advantage is the generalization that spatial data gives, so that you don't have the masks to match in resolution. They don't have to match in resolution the image. They can be different. One can be polygons, one can be an image, one can be a different representation. Still, the method will compute the tiles for you. And last two slides, spatial muon actually supports also graphs, because I was applying also some graph neural networks. So you can build graphs from spatial data, and we still don't have this in the agenda for the new structured version, but at some point I think it will come. And finally, we use this also to store some more exotic types, like tomosic data, in which you have a sample that is sliced in different regions, and each region has got an expression vector. So you want to have, instead of having one image and a set of coordinates, you want to have one object that allows you to make the plot on the right. And spatial muon did this by computing the alignment to space automatically. The scale bar was computed automatically and allowed us to make complex plot like the one on the left. So you have 4 different layers of information that on the right, this is what is actually computed. So you see that the alignment is non-trivial. This is very time consuming if you have to do it by hand, but then it was done automatically. So by just selecting the common region, you will get the line plot on the left. So this was everything. So I want to conclude by saying that we propose we are developing 3 plugins. The first one being public will be spatial data, and then a Napari plugin and a plugin for multi-modal analysis that will come later. And for doing this, we are proposing an extension to the open source standard OME-NGFF. I want to thank all the people involved with the project. On the left, there are the people from my team that contributed with spatial muon and now are contributing for spatial data and spin. On the right, there are collaborators from the Theis Lab and from Dagmar Iber Lab from ETH, and ETH and Helmholtz, and these are helping me with the spatial data, and helping them with the Napari plugin. And finally, there are many collaborators that were kind to provide data and give analysis and discussion that really help to shape the development of these projects. Thanks for your attention, and I'm happy to accept questions. Thank you.
Yeah, wow, a lot of stuff. Thanks a lot. So maybe just shout out if somebody has a question.
Yeah, I'll start. Thank you, Luca. This was a fantastic talk. Really, really great visualizations and workflow here.
Thank you.
I wanted to ask first, what you're putting together several software packages, and I was wondering what the interface to all this was. I think you were showing us some code at the end here, and this looked like it was maybe coming from a Jupyter notebook. Is that the main way that you would expect users to interact with this, or are you thinking of going for a complete graphical interface with Napari? Kind of what do you have in mind, how to tie it together for an end user?
The interface is actually a bit even more low level than Jupyter. You can use it from Jupyter, but it's designed also to be used for method developers. Like if you do deep learning, maybe you are used to write I don't know, Python files and not Jupyter notebook files. So it's designed to be like a package, like a standard Python package. But then, as I said, you can use it from Jupyter, and the graphical interface is only for the analysis part. So if you do an analysis of a spatial multi-omics data set with Jupyter files, then at certain points, maybe you want to align different layers. And at that point, you can call the Napari plugin work on that and then get back to Jupyter. This is what is being done now with SquidPy and what we want to keep supported.
Okay, and this Jupyter Notebook, where do you usually run it? Is it just on your local laptop or is it on a HPC system?
So personally, you run it also either from my laptop, either from the cluster. You open a tunnel connection with the graphic interface forwarding. Yeah, like so with ZARR, there is the possibility to support cloud. We haven't looked into that yet, but yeah, maybe having that, you could also think that you could run the notebook in more like the task ways, but we haven't thought about that. So let's say local machines or SSA's tunnel forward access to remote machines.
So right now the OME-ZARR is being saved to a file system as opposed to S3 or something like that. Okay.
Yeah.
Hey, I have a question. What about 3D data? Most of what you showed is 2D.
Yeah.
What about 3D data? And another question is, in Python, you know, NumPy has this like array abstraction, or I don't know how to call it. I'm not a programmer, but there's like some, you know, Dask and NumPy work really well together, but also X-array, which is supposed to give you the spatial coordinates on top of NumPy Dask back things.
Yeah.
If you could comment on, you know, differences between spatial data and X-array.
Yeah. So I start with the first question, 3D data. So the development of 3D data is in our mind, but not in the agenda yet. This means that when we develop some structure for the data, we maybe like put like not implemented to do, so that we know that we will have at some point to fill that block. But we are focusing more on like visualization. So with SquidPy, you can already plot 3D data, and it's quick to implement the plotting. For the storage, we know that we don't have to write something that's just for 2D data. So that's our way. Actually, for 3D data, there is another building blocks that I haven't mentioned. It's called we call it mesh regions, volumes more specifically, and it's again drafted in the code, but for a moment we don't have time to implement it. Regarding the second part, yes, X-array is a great way to map the coordinates for its layers of the images, and we are considering using it. Like SquidPy works by using enriched and data representation. So you have and data with some spatial information plus a class that is an image container that is a bridge. Is basically an image that is bridged to the data object. And inside there, X-array is used. So we are considering if using that or porting the old spatial muon code in which we are not using that. And the reason is that X-array works for raster data for tensors, like NumPy tensors, but sometimes, but you want also this physical information to be valid also for regions and for single molecule. So we are seeing what's the cleanest way to make it work. The old spatial muon code, we are not using it.
Yeah, because the combination of two things is what troubles me, right? Once you get to meshes and 3D, you can't have in-memory representation because the data could be terabytes,
yeah,
right? And then it helps to just be backed by Dask and not worry about it.